Hello and welcome everyone to this video podcast for evolution and diversity. This is our fourth and final video covering bacteria and archaea. And in this video we're going to talk about what is sometimes referred to as the Great Oxygen Revolution. Then we're going to talk about key lineages of bacteria and archaea. And then I'm going to close with a very brief description on a couple ways that we study bacteria and archaea. So let's go ahead and start talking about this Great Oxygen Revolution. Now I'm going to show a video that I think explains the Great Oxygen Revolution in a much better way than I could draw it out for you. As you're watching this video, I want you to think about some questions or just some general themes. And I'm going to list those things right now. And the first one is, what were the oxygen concentrations like on early Earth? And then I want you to think about cyanobacteria again. Cyanobacteria keeps popping up. It turns out to be an incredibly important bacterium. For this point, though, I want you to think about its adaptations that led to the oxygen revolution. And next, how cyanobacteria and its ability to make oxygen led to the first and perhaps the longest ice age on Earth. The next question I want you to think about is, how did this allow for the explosion of aerobic organisms on Earth? And lastly, how did our good friends, the cyanobacteria, popping up here again, help lead to the formation of plants? And this will be a good stepping stone into our next unit of material where we're going to spend quite a bit of time talking about plants. So let me go ahead and move to that video.
There's an organism that changed the world. It caused both the first mass extinction in Earth's history and also paved the way for complex life. How? By sending the first free oxygen molecules into our atmosphere. And they did all this as single-celled life forms. They're cyanobacteria, and the story of these simple organisms that don't even have nuclei or any other organelles is a pivotal chapter in the story of life on Earth. Earth's atmosphere wasn't always the oxygen-rich mixture we breathe today. 3.5 billion years ago, the atmosphere was mostly nitrogen, carbon dioxide, and methane. Almost all oxygen was locked up in molecules like water, not floating around in the air. The oceans were populated by anaerobic microbes. Those are simple unicellular life forms that thrive without oxygen and get energy by scavenging what molecules they find. But somewhere between 2.5 and 3.5 billion years ago, one of these microbial species, probably floating on the surface of the ocean, evolved a new ability: photosynthesis. Structures in their cell membrane could harness the energy from sunlight to turn carbon dioxide and water into oxygen gas and sugars, which they could use for energy. Those organisms were the ancestors of what we now call cyanobacteria. Their bluish color comes from the blue-green pigments that capture the sunlight they need. Photosynthesis gave those ancient bacteria a huge advantage over other species. They could now produce their own energy from an almost endless supply of raw ingredients. So their populations exploded, and they started polluting the atmosphere with a new waste product: oxygen. At first, the trickle of extra oxygen was soaked up by chemical reactions with iron or decomposing cells. But after a few 100 million years, the cyanobacteria were producing oxygen faster than it could be absorbed, and the gas started building up in the atmosphere. That was a big problem for the rest of Earth's inhabitants. Oxygen-rich air was actually toxic to them. The result, about 2.5 billion years ago, was a mass extinction of virtually all life on Earth, which barely spared the cyanobacteria. Geologists call this the Great Oxygenation Event, or even the oxygen catastrophe. That wasn't the only problem. Methane had been acting as a potent greenhouse gas that kept the Earth warm. But now, the extra oxygen reacted with methane to form carbon dioxide and water, which don't trap as much heat. The thinner atmospheric blanket caused Earth's first, and possibly longest, ice age, the Huronian glaciation. The planet was basically one giant snowball for several 100 million years. Eventually, life adjusted. Aerobic organisms, which can use oxygen for energy, started sopping up some of the excess gas in the atmosphere. The oxygen concentration rose and fell until eventually it reached the approximate 21% we have today. And being able to use the chemical energy in oxygen gave organisms the boost they needed to diversify and evolve more complex forms. Cyanobacteria had a part to play in that story, too. Hundreds of millions of years ago, some other prehistoric microbe swallowed a cyanobacterium whole in a process called endosymbiosis. In doing so, that microbe acquired its own internal photosynthesis factory. This was the ancestor of plant cells, and cyanobacteria became chloroplasts, the organelles that carry out photosynthesis today. Cyanobacteria are still around in almost every environment on Earth: oceans, freshwater, soil, Antarctic rocks, sloth fur. They still pump oxygen into the atmosphere, and they also pull nitrogen out to fertilize the plants they help to create. We wouldn't recognize life on Earth without them, but also thanks to them, we almost didn't have life on Earth at all.
So in that video, you could see how cyanobacteria producing all this oxygen ultimately led to a lot of death and destruction, but in the end, it was also responsible for generating the diversity of life that we see now and that we see in the fossil record. And so what I'd like to do now is spend a little time talking about some key lineages of bacteria and archaea. I'm going to begin with bacteria, and there are 6 of these lineages of bacteria I want to talk about. I thought I would show slides for these because it's much easier to see the pictures, and I'm going to highlight what I want you to know from each of these slides. And if you want to look in your textbook, these are also summarized in tables 26.4 and 26.5. So let's look at this first lineage of bacteria that you need to know. And these are called the actinobacteria. And as you'll see, there's a lot of information listed here about actinobacteria. But all I want you to know about actinobacteria is that some of these species are linked to causing tuberculosis and leprosy. You should also know that members of this lineage are responsible for producing many of the antibiotics that we humans use in our modern medicine. The second lineage is chlamydia. What I want you to know about chlamydia is that all species of chlamydia, they live as parasites. And some of these cause sexually transmitted diseases, like chlamydia. The next lineage of bacteria, cyanobacteria, we've talked about several times during the past several videos. And so there's a lot that you need to know about cyanobacteria. So if we've mentioned it before, you should make sure you remember it in the context it was mentioned before. But some of the key highlights of cyanobacteria include what we just talked about, and that is how they are responsible for the oxygen revolution. They're also responsible for the Great Extinction that wiped out almost all forms of life. They're also responsible for the first ice age, yet they are also responsible for the diversity we see on Earth today. So they've had quite a mixed past. So they're a very interesting bacterial species. You should also remember that some of these cyanobacteria are important in nitrogen fixing, and they are photoautotrophs because they are able to undergo photosynthesis. The next one I want you to know is Firmicutes. And what I want you to know about this lineage of bacteria is that they're found in the human gut. However, they can also be a pathogen. They are linked to anthrax, botulism, tetanus, gangrene, and strep throat. And interestingly, even though they're linked to all of these specific pathogenic diseases, they can also be used to make yogurt and cheese. So there's good and there's bad with this group. Okay, this lineage of bacteria are called the proteobacteria. This is the one you've probably heard of the most because it represents E. coli, and most people have heard of E. coli in one form or another. We know that these bacteria are found in aquatic environments, and some of them are pathogens. And we've talked about this before when we talked about pathogenic bacteria, but remember that some forms of E. coli are linked to cholera, food poisoning, plague, and dysentery. Now, the last lineage of bacteria that I want you to know are the spirochetes. These are found in the guts of animals. They are also pathogenic bacteria. They are linked to diseases like syphilis and Lyme disease. As you can see in this picture, they have this very interesting corkscrew shape. This corkscrew shape allows them to burrow deep into the tissues of their hosts. Okay, next I'd like to talk about just 3 lineages of archaea. Okay, this first lineage of archaea that I'd like you to know are the Crenarchaeota. You should know that species of Crenarchaeota are found in extreme environments that include very high heat, high pressure, and very acidic environments. They also represent the smallest of all archaea and bacteria. Okay, the second lineage of archaea I'd like you to know are the Euryarchaeota. These also live in pretty diverse habitats. We find them in the human gut as well as the guts of other animals. And we also see them in hydrothermal vents and some abandoned mines. They're well known for the ability to produce methane byproduct. In fact, they add billions of tons of methane to the atmosphere each year. Okay, the third and last lineage of archaea that I'd like you to know are the Thaumarchaeota. Members of this lineage are found in a variety of habitats, including freshwater, saltwater, soil. They are probably the most abundant in oceans. You should also know that this lineage will use ammonia as a source of energy, and in that process they produce nitrites as a byproduct. Okay, so we just went over 6 lineages of bacteria and 3 lineages of archaea. So make sure you know these different lineages and be familiar with the information that I told you about each one of them. And I wouldn't focus so much what's in the table, but focus more on what I included as the brief summary for each of them. I haven't written the exam yet, but I would imagine that this would make a really good matching kind of a question. Okay, we've spent the past several videos talking about the many adaptations that bacteria have, why they're important for us to study, how they impact the environment. We talked about their evolutionary significance, and we just got done talking about the many different lineages and variety of bacteria that we see on Earth. So now what I'd like to do is just spend a little bit of time talking about how we study bacteria. And we could spend an entire course thinking about and talking about how we study bacteria. And I'm only going to talk about a couple different ways that we study bacteria. Okay, so let's go ahead and get started on these couple ways we study bacteria. All right, so how do biologists study bacteria? The first is using something we call enrichment culturing. And the second is called metagenomics. Let's focus on enrichment culturing first. So this is a very traditional way of studying and identifying bacteria. And so you would take a petri dish with a solid media surface on it, and this media would have different types of growing conditions. It may vary in temperature, salt concentration, lighting, the type of substrate that the bacterial cells would grow on. The nutrient source could be different kinds of sugars, different kinds of amino acids, different kinds of vitamins that could be added or removed from this agar. By changing these different conditions, a biologist can find the optimal temperature, can find the optimal conditions that certain bacteria grow on. So let's take a view of this plate looking down on it. So I'm going to just draw it like this now. A scientist may go out into the environment, maybe grab a soil sample, maybe samples from a tree or a plant, or maybe samples from an animal, from the ocean, wherever it might be. And they will plate it on petri dish here. And they may have several of these plates that they've plated the same bacteria on, each with a different set of conditions, trying to identify what conditions will allow certain bacteria to grow. And so what they may find is that under some conditions, maybe you see more colonies growing. Maybe under different conditions, you'll see colonies growing, but maybe they're different kinds of species, and so on. You may have some conditions that don't support any kind of growth. And so this is really useful in identifying bacteria that grow in certain environments, because we can take these bacteria off and we can study them. We can study them in a variety of different ways. But this is where the problem comes, and that is this only identifies culturable species. And what I mean by that is maybe under these environments here, there are actually bacteria here that were pulled from the soil, the ocean, or whatever environment it was that you took the sample from, but they are not able to grow In fact, you wouldn't even see this dot I'm making here. I'm just making it so we can see it in this example. But in reality, when you look at the Petri dish, you would never see anything. But they're there. They're just not culturable. We cannot find conditions, there are not conditions that we know of that allow these to grow in a laboratory. But they still exist, so there has to be a way to identify what these unculturable bacteria are, and archaea. And that's when we come into our second way to study bacteria with metagenomics. So what is metagenomics? This is the study of all genetic material within an environmental population. So how does this work? Well, let's say that a biologist brings in from the field a sample of soil. We know this sample of soil has thousands of different species of bacteria and archaea in it. We know if we culture this on a petri dish, we're not going to identify all the species in this soil. We're going to miss a lot because not all bacteria or archaea are culturable. So we're going to take samples of this soil and isolate the DNA from all the microorganisms. And then we sequence this DNA. And then we compare it with known species and identify differences within these common genes. And doing this, we get a much better picture of the diversity of different microbes within this population. And it's revolutionizing our understanding and appreciation of the diversity and abundance of microbes in our environment. Okay, let's move on to our last topic for this video podcast, and that is the topic of the human microbiome. The field of metagenomics has expanded our understanding of the many different bacteria and archaea and other microbes that live in different environments. One of those environments that we're very interested in is the human environment. So the human microbiome is a very specific study of metagenomics. But let's give the human microbiome a little bit better definition. So the human microbiome encompasses all the microbes that inhabit the body or parts of the body. This includes our digestive tract, respiratory tract, parts of the reproductive tracts. You'll also find these microorganisms in your ears and eyes, your skin, and they are also found in breast milk. We find bacteria in most parts of our body, and these are just a few examples. And it should be noted that the vast majority of microorganisms that live in our body that are part of this human microbiome are part of the digestive tract, and most of them are in our gut, but they play important roles in other parts of our body as well. Oh, and before I go any further, I should point out that when I say microorganisms, I mean all kinds of microorganisms, but the vast majority of them are bacteria, and archaea would also include quite a few of these microorganisms. As technology has developed and we're learning more about these bacteria and archaea that live within our bodies. And remember, I told you earlier that bacteria and archaea, their numbers in our body is much more than all of our cells combined. So they're a very large part of our body, and we're learning that they are very important in human health. We're also learning that depending upon the types of microorganisms that make up the human microbiome, they can be linked to things like obesity, diabetes, that is type 2 diabetes, irritable bowel syndrome. The microorganisms that make up our human microbiome are also linked to certain kinds of behaviors, such as ADHD and even autism. Now, I want to be clear that I'm not saying that the microorganisms in our body are directly and the only things that can lead to ADHD and autism. They are just a factor that helps explain these complex behaviors. There's also some evidence that the human microbiome contributes to cognitive development. And really the list goes on and on of everything that has been linked to the human microbiome. And as I said before, it doesn't mean it's the only thing that is causing these various health-related syndromes, but it is a factor that we're learning contributes to them, and in some cases may contribute largely to them. Now, the last thing I want to mention to you today in regards to bacteria and archaea are the various microbiology courses that you can take at CMU. The first course I'll mention is Biology 208, and this is simply called Microbiology. It's a very good course to take. However, it is not designed for biology majors, and it won't count towards your biology degree. So if you're going to become a biology major, this is not the course that you'll take. There's another course that'll be more appropriate for a biology major. However, if you're not going to be a biology major, which I hope you will be, but if you're not, and you want to learn more about microbiology, this is a course that you might want to take. The next course I want to mention to you is Biology 320, and this is called Biology of Microorganisms. Now, this is the microbiology course that you take if you're a biology major. So if you want to learn more about microbiology, once you're done with the 5 core courses of our program, then you can take Biology of Microorganisms. Biology 539. This course is called Virology. Now, this course deals, as you might suspect, with viruses, and we haven't really talked about viruses yet. We've spent most of the past several videos talking about bacteria and archaea, but it is a course that I think relates quite well with a lot of the topics we've talked about over the past few videos. The next one I want to mention is Biology 565, and this course is called Microbial Diversity and Physiology. This is a really good course to take after you've taken Biology of Microorganisms. And the next course I want to mention is Biology 580, and this is called Medical Microbiology. As the name implies, this course is going to spend a lot of time talking about the medical applications of microbiology and spends a lot of time talking about pathogenic microorganisms. And the last course I'm going to mention is Biology 585. And this course is called Conservation Medicine. Now, it's not a course that is exclusively about microorganisms, but a very large part of this course deals with pathogenic microorganisms and how it is related to wildlife, domestic animals, and humans. It'll focus on the epidemiology and ecology of these diseases that are transmitted by microorganisms. So these courses will cover a lot of microbiology within their content. There are other upper-level biology courses that will also touch on microorganisms and microbiology, but these are the main ones right here. If you have questions about these courses, please feel free to ask me, and I'll be happy to help address any questions you might have. Now, these courses you won't be able to take quite yet. You'll have to wait until you finish the first 2 years of the biology program, but then you should be able to fit in a few of these if you would like to. Okay, that is all I have for today. If you have any questions at all, please make sure you come see me. If not, I will see you in class. Bye for now.